Assalamualaikum students. Before going to our educational topic, please subscribe my channel Muntzir Mehdi Science TV on YouTube. Press the like button and share this lecture to your friends and classmates who have no access to it to add in their science education. And those students that are preparing privately can benefit from our channel very effectively. Let's come back to our topic, that is recombinant DNA technology. We can also write it as RDT. First we see its definition, then we would explain it with the help of theory and diagrams. Recombinant DNA technology, also known as genetic engineering. Its definition is such a DNA that consists of a single DNA from two different organisms to different sources is known as recombinant DNA, and that technology which is used for the preparation of recombinant DNA is known as recombinant DNA technology. You must memorize that during the formation of recombinant DNA, two organisms are involved. The first organism may be a human, but the second organism either a bacteria or some other organism. Also suppose recombinant DNA technology with the help of a diagram. Let's suppose the first is a human cell. At the center there is present DNA, whereas the second cell can be a bacterial cell or some other organism's cell. It is the plasmid of bacterial cell, whereas it is its DNA. For the preparation of recombinant DNA, a small gene piece is taken from the human chromosome and plasmid is cut out from the bacterial DNA. At the next stage, both human gene and the bacterial plasmid recombine and it becomes a recombinant DNA. So now students, you have learned how to make a recombinant DNA. Next, we shall see different materials which are required for recombinant DNA technology. The first one is gene of interest is to be cloned or inserted. We can see here it is our gene of interest which is to be cloned or added. Next is a molecular scissor that is used to cut out the gene of interest and the bacterial plasmid. It not quite matched to that scissor that commonly use our mothers for cutting cloth, but this molecular scissor is in the form of an enzyme. Third material which is required for the recombinant DNA technology is molecular carrier or vector, on which gene of interest or gene of choice could be joined, could be placed. At the fourth step, the gene of interest along with the vector, gene of interest completely joined with the vector like this. When it is introduced in an expression system to produce a specific product. Next, we will see how recombinant DNA technologists get a gene of interest or gene of choice. For obtaining gene of interest, three possible ways are present. The first one is isolation of gene from the chromosome. That is a simple and common method for obtaining gene. In this method, genes can be obtained from the chromosome by splitting the chromosome on the flanking side of the gene by using a special enzyme that is known as restriction endonucleases enzymes. That is the similar enzyme present in the form of a molecular scissor. Second way is the chemical synthesis of genes. In it, if genes are of minimum size, then they can be easily synthesized in the laboratories. Third is preparation of gene from the messenger RNA. That is also known as complementary DNA. It is most common method of getting gene in this. The gene is prepared in the laboratory from the messenger RNA strand. During the process of reverse transcription, this process an enzyme is used that is called reverse transcriptase enzyme. The DNA molecule is known as complementary DNA. Next, we will study about molecular scissors that is in the form of restriction endonucleases enzyme. These restriction enzymes are naturally present in the bacteria. These bacteria use them for their own protection, own safety against viruses, because viruses can enter the bacterial cells and harm their bodies and also infect them with different diseases. In bacteria, the role of restriction endonucleases enzymes is that these enzymes cut down the viral DNA but do not harm to the bacterial chromosome, as these enzymes restrict the growth of viruses. So these are known as restriction endonucleases enzymes. First restriction enzyme is isolated by Hamilton D. Smith, scientist that was a biologist in 1970 when he was working at Johns Hopkins University. Now then, more than 400 restriction endonucleases enzymes have been isolated, out of which 20 are frequently used in recombinant DNA technology. So these restriction enzymes can cleave the human gene and plasmid DNA from the middle. Restriction endonucleases enzymes means they are restricted to cleave at the specific point only. Usually these have specific recognition pattern where exactly these will cleave, and those patterns are mostly known as palindromic sequences. Palindromic sequences are the specific sites present on the DNA molecule. These specific sequences consist of 4 to 6 nucleotides that are arranged symmetrically in the reverse order. EcoR1 is the special restriction enzyme which detects and responses to the sequence such as T88TG, and whenever it finds this sequence such as GTTAAC, it is called the palindrome because whenever it reads forward and in reverse direction it will be the same. That's why it is known as palindrome. So all the restriction enzymes first detect a specific palindromic sequence and then they cleave at a specific point. Now EcoR1 recognizes this specific pattern of palindromic sequence but cleaves it right after GA. So it will cleave the DNA like this fashion, like this pattern. It is an example of endonucleases enzyme and palindromic sequence. So this restriction enzyme will be applied to cleave this segment out. So now this DNA segment that is called a human gene lives from here. Now our target gene has been cut out. Now what we need to do for this target gene? Now see, for its insertion, we need to cut out the plasmid vector and also treat it with the same EcoR1 enzyme so that we can create the palindromic sequence like this. Here it is necessary to keep in mind that the target gene and the vector plasmid are needed to treat with the same EcoR1 enzyme so that the creation of palindromic sequence is possible. Otherwise, if different enzymes are used for cutting the target gene, we use EcoR1, and for vector plasmid BAMH1 we use, then palindromic sequence will not be created and recombinant DNA will not be prepared because complementary match of base pair will not be occur. So for the complementary match of nitrogenous bases of the DNA or RNA nucleotide, same restriction enzymes would be used. In this case, EcoR1 is used to cut the target gene out, and also the same EcoR1 enzyme is also used for cleaving the vector plasmid. So when both of these segments have been cut, then question comes in our mind, how will these two segments, the target gene and the vector plasmid, be joined again with each other? For this purpose, the special enzyme is used that is called the DNA ligase. DNA ligase is that particular enzyme which can seal the nicks of the target gene and the vector plasmid that consists of palindromic sequence. DNA ligase enzyme do not add any additional nucleotide, but it only joins the nicks. So DNA ligase acts as a glue. We can say that restriction enzyme is a molecular scissor whereas ligase acts as a molecular glue. So now we have constructed the target gene into the vector plasmid. This construction, this combination is known as recombinant DNA technology. I hope you are clearly understand the big picture of recombinant DNA technology. Students, strictly keep in mind that EcoR1 is the restriction endonucleases enzyme that is used to cut the double-stranded DNA molecule that consists of target gene right here that consists of particular palindromic sequence of nitrogenous bases at the cleavage site, whereas on the other hand the same restriction enzyme EcoR1 produces a gap in the vector DNA, and a piece of foreign DNA with its complementary ends like this can be placed in this gap. These two separate DNAs but that are present singly, this and this. So the complementary ends of these two DNA molecules are known as sticky ends because they can bind by complementary base pairing. The restriction enzyme EcoR1 help the insertion of foreign DNA into the vector DNA. We can also understand it with the help of another diagram. Suppose these are the sticky ends of the target gene, whereas these are the sticky ends of the vector DNA. So EcoR1 cuts the specific sequence of nucleotides in such a way that these sticky ends are produced. When they join with each other, then recombinant DNA is produced. Next, we will see the molecular carrier, that is also known as vector. As we have known that for synthesizing the recombinant DNA, vector molecule is also necessary and it is selected. A vector is a carrier molecule that is cut like this, and then target gene is inserted into it. After the insertion of target gene, it becomes a recombinant DNA, and finally it is introduced into a host cell. Most commonly used vector is plasmid. Plasmids are natural extra chromosomal circular DNA molecules present in bacteria that were discovered by some investigators that were studying the sex life of the intestinal bacterium E. coli. Antibiotic resistance and fertility genes are present on the plasmids. PSC101 and PBR322 are two known plasmids. PSC101 is earlier discovered plasmid, first plasmid vector used for gene cloning that has antibiotic resistance genes for tetracycline, whereas PBR322 is also first widely used E. coli cloning vector which has antibiotic resistance genes for tetracycline and ampicillin. Both these plasmids provide site for the insertion of gene of interest. At the second point, DNA of bacterial virus can also be used as a vector. For example, Lambda phage. Vector selection depends upon the gene of interest. Which type of gene of interest will be their matching vectors would be selected, either a plasmid or a Lambda phage. So at the end, we will use the insulin gene and understand the complete formation of recombinant DNA and then its introduction into the host bacterium by using a diagram. Suppose it is a human cell. At its center, there is present DNA. So from this human DNA restriction, endonucleases enzymes such as EcoR1 cuts an insulin gene that has palindromic sequence. On the other hand, a bacterial cell consists of plasmid DNA. From this plasmid DNA, a piece is cut with the help of same restriction enzyme. The enzyme is same which is used to cut the gene of interest. And this gene of interest, that is insulin gene, is joined with the sticky ends of the plasmid with the help of the enzyme DNA ligase. DNA ligase seals the foreign piece of insulin gene into the vector plasmid so that DNA from two different organisms have joined together, which is now called recombinant DNA or chimeric DNA. Bacterial cells take up this recombinant plasmid if they are treated with calcium chloride solution to make their membranes more permeable. So after the treatment of calcium chloride, recombinant plasmid is taken up. Then cloning occurs. This bacteria reproduce and bacterial clones are formed. Each new bacterial cell contains at least one plasmid. Therefore, each bacteria contains the gene of interest which will express itself and make a product. From these bacterial clones, cloned genes can be isolated for further analysis and protein products such as insulin can be separated. This is the whole mechanism of cloning of a gene such as insulin gene. So a clone can be a large number of molecules such as cloned genes or cells such as cloned bacterium or organisms that are identical to the original organism. Students, at the end I will advise you keep your focus to your education. Doesn't only benefit you but your society, your family and also your country. Thank you for watching this your video.